Hi, good morning, everyone. Let's continue our discussion about how cells utilize energy and how that involves the production of ATP. So we're going to shift our conversation to look at photosynthesis. In other words, how do cells in organisms like plants, for example, actually take the energy from the sun to make ATP? And along the way, how does the production of glucose fit into that? Now, I remember when I was an undergrad, I kind of dreaded learning about photosynthesis because, well, two things. It was really complex sounding, and it was just about plants, right? And I like plants. I love plants. I grow plants at home, things like that. But it seemed like, well, it's not really relevant to me understanding photosynthesis if I wanted to go on and study things like, human health and how cells in our body work, right? But I want to try to encourage you to think about it this way. One of the big themes in our Bio 275 course all semester has been trying to appreciate how all life on Earth is tied together, right? And how, because we all come from the same original ancient ancestral first life on Earth, right? And as life evolved, it had to go through many different changes in terms of what was available. Raw materials, things like oxygen, that led to things like aerobic respiration. I think it's a good opportunity to look at how photosynthesis works as a very critical part of the story of life on Earth. You would not be here if it were not for the photosynthesis that's going on right now, right, in the plants outside of your window. So let's take a look at that. First I wanted to start off with a little story here. So I want to show you a picture of a place. So this is Cutler Pond, which is in Newton, Massachusetts. So I grew up in the town of Needham, Massachusetts, and my home was about, like a less than 10-minute drive away from this pond. It was a wonderful wooded area, and this pond wasn't that big. I think if you were to walk the trail that goes around the pond, I think it's about something like a 3 mile all the way around through the woods and along the shore. So as a kid, my dad used to take me to this pond and we'd go fishing. And so I have lots of fond memories of sitting there with my dad along the banks, fishing during the summer. And we'd catch things like these little sunfish, or oftentimes I'd hook a catfish, right? And those catfish, they're like all muscle, right? So I remember as a kid, I'd be trying to reel this thing in, and I was convinced that I caught something like a whale, right? But then it turns out that it's just like this catfish at the end, and then, often we just let them go, or sometimes we'd take them home and we'd fry up fish for dinner, right? Also, as a kid in elementary school, we used to take field trips to this pond to learn about things, like ecology and appreciate living things, as kids. We also caught poison ivy there. Boy, I caught poison ivy a lot from there. We don't have poison ivy here in Hawaii, and that's probably a good thing. Anyhow, so this is a place that's very special to me, and this is how it looks during the summer, like in June or July. All right. So later on, as an adult, when I went back to Boston to do my postdoctoral work, I often very fondly would come and visit this lake, and I'd run around it, things like that. Right? So this is what it looks like during the summer, but now we're in November, right? So in the fall, it looks quite different, right? This is what it looks like there right now. All right, so growing up in this environment, I never fully appreciated how wonderful it is when the seasons change and the leaves change color, right? What was bright green during the summer became these wonderful blazing yellows and oranges and reds in the fall, right? And of course, this change of the colors of the leaves, right, is a normal part of how foliage evolved in places where you have dramatically changing photoperiods and temperatures throughout the year. So I never fully appreciated this as a kid. But when I went back as an adult, right, and I would be running along this path, oftentimes in the evening, looking out at these orange and red leaves, it really made me think about the fact that how many years had kind of gone by, right, since I was a little kid there fishing with my dad or going on field trips with my classmates, right? And all those years, these living things, these trees, these plants had gone through this yearly cycle of growing green leaves that would then turn yellow and orange in the fall and then fall off during the winter, right? So it really made me think about the passage of time and how lots of times we get so caught up in our daily routines that we don't stop and think about how life around us kind of keeps going and going. All right, so we'll talk about the changing colors of leaves in a little bit, but this was sort of my introduction to photosynthesis. Right? So what is photosynthesis? We already introduced photosynthesis in a very sort of, superficial way this semester when we talked about how photosynthesis is when certain organisms can take the energy from the sun, right? So the energy from the sun represents all the energy that fuels life on Earth. And certain organisms can take water from the air, carbon dioxide from the air, right? And combine or recombine those atoms, those carbons and oxygens and hydrogens, right, to actually store the energy of the sun in carbon-carbon bonds as molecules of glucose. So previously, I think most of you are familiar with a cartoon that would show the sun shining on a leaf and water and carbon dioxide having arrows going in, and then you have glucose coming out, right? And you have oxygen as a waste product of that. Right? But what I'm going to tell you now is that photosynthesis actually takes place as two sort of discrete and separate processes. Those two processes are broken down as energy transduction. In other words, the steps of taking the energy from the sun, right, and using water to make ATP. So in other words, this is directly making ATP using the energy of the sun. Okay? And then you have oxygen as a leftover product. The second step is what we call carbon assimilation. And what that means, to assimilate means to kind of pull together or to become, joined together. And what happens there is the plant takes some of the ATP that it made from the first step over here on the left and takes that energy from the ATP, right, to convert carbon dioxide into sugar, right? So the net reaction then, if you combine these two together, is that light plus 6 molecules of water plus 6 molecules of carbon, right, across these two different parts of photosynthesis will yield 6 molecules of O2 as waste, and importantly, a molecule of glucose, right, of sugar. So you might think to yourself, wait, if the whole point of making sugar is just to break it back down to ATP, right, like what our cells do, then why does the plant even need to do this, right? If it can already make ATP directly by harnessing the energy from the sun and making it into ATP? Yeah, that's a good question, right? Why go to the trouble of making sugar if you're just going to break the sugar down again? It all has to do with the fact that if you compare the cartoon on the left with the cartoon on the right, what is missing in the cartoon on the right? The sun, right? Because think about it this way. If the sun is driving the direct production of ATP on the left, what do you do if there's no sun, right? And I don't mean the sun just suddenly like blows up and disappears, because then we got bigger problems to worry about. I'm just talking about, what do you do when the sun goes behind a cloud, right? Or even more obviously, when it's nighttime. Do living cells and plants not need ATP at nighttime? No, of course they do. Well, if it was dependent entirely on the availability of sun to make those ATPs, well, to put it mildly, the plant would kind of be screwed, right? So that's why plants make ATP and store some of that ATP by converting it into sugar, right? So that literally saving the ATP for a rainy day, right? Or every night, it at least has sugar to break down back into ATP. Consider also that not all the tissues of the plant are the leaves that actually do photosynthesis, right? The tissues inside the stem or the trunk of the plant or the cells that are in the roots of the plant, they don't do photosynthesis, but they need ATP. So when the leaves make sugar from some of the ATP that's made from photosynthesis, that sugar can then be moved around in the plant so that those other cells can take the sugar and break down that sugar through glycolysis and aerobic respiration, just like what we do, right? And of course, it's worth it to mention that how do we fit in this picture, right? We come along in this picture, right? We sort of like, kind of like, sort of ruin things for the plant here because we can't photosynthesize ourselves. We evolved to purely take the sugars that the plants made, right, and eat them. And then that's how we get the sugars to power our cells. Okay. All right. So I want you to notice that the H2O and the CO2 are actually involved in different parts of photosynthesis. And I want to remind you again to always keep in mind that photosynthesis is essentially the reverse of glucose oxidation. That if you start with a molecule of glucose and then proceed to fully oxidizing it using 6 molecules of O2, you end up with, right, the 6 molecules of carbon dioxide and the 6 molecules of H2O that essentially went into photosynthesis, right? So it's that wonderful cycle between us and the plants, right? The plants make the sugars and give off the oxygen, right? And then we need those sugars and the oxygen that the plants made, right, in order to fuel our cells. And in return, we breathe out H2O and carbon dioxide, right? That becomes the raw ingredients again for the plant to make more sugar through photosynthesis, right? By taking more sunlight to make the glucose molecule again. All right, so where does this take place inside of a green leaf of a plant? Inside of a leaf, let's zoom in here, you'd have these plant cells, right? So plant cells are eukaryotic cells, just like ours. Right? So they have all the same types of organelles that our cells have, a typical eukaryotic cell. But in addition, you'd find this extra organelle, the chloroplast, right? So inside of these cells, you'd find these green sort of shapes here. If you were to zoom in and look at it sort of on the side, it's sort of like a stack, another example of something that looks like a stack of pancakes, right, of membranes. And this is the chloroplast. Each one of these little stacks inside of it is what we call a thylakoid, right? So it's a series of membranes that are basically stacked on top of each other. And just like when we talked about stacks of membranes and different functions that they perform in things like the endoplasmic reticulum and Golgi, right? You should already be aware, it's no accident that you have stacks of membranes that perform some sort of important function. Right? So this chloroplast is what you'd find in sort of a proper plant cell, right? But of course, life on Earth did not start off with just all of a sudden having a plant with leaves like this maple leaf here. I choose the maple leaf because it kind of reminds me of being a kid on the East Coast. Early life that was photosynthetic, right, was prokaryotic. It's just a single-celled organism. So something like this. Early cyanobacteria were photosynthetic. Remember, I showed you I showed you a nice artistic painting of what early life looked like when you had the ocean, right? And maybe the sky was filled with carbon dioxide, and in the ocean you had early photosynthetic bacteria, right, that were busy harnessing the energy from the sun, plenty of water, plenty of carbon dioxide, and giving off little bubbles of O2 as waste products. Right? So these photosynthetic prokaryotes, they don't have chloroplasts, right? Because they're themselves almost like an actual chloroplast themselves, right? They're like free living chloroplasts. You can think about them that way. Just like mitochondria, we now believe originally started off as separate little organisms, prokaryotes, right? Mitochondria and chloroplasts at some point in ancient evolution of eukaryotic cells, once cells started to develop different organelles like a nucleus, and things like chloroplasts and mitochondria kind of joined up with them, right? Because you had a symbiotic relationship where the little photosynthetic prokaryotes or the little mitochondria benefited from being able to hang out protected inside of a larger cell. And then that larger cell benefited from having these little guys inside of them because the chloroplasts and the mitochondria were producing good stuff, right? They were producing energy that the big cell benefited from. And over time, that relationship became inseparable, right? And that's why our eukaryotic cells in our body today, right, have echoes of that ancient past. Now, we don't have chloroplasts, but we certainly have mitochondria. And you may be surprised to know that plant cells not only have chloroplasts in them, but they also have mitochondria. Okay. For some reason, a lot of students think that plant cells have chloroplasts instead of mitochondria. But once we discuss photosynthesis and you suddenly realize that plant cells need to use glucose as well, then of course it seems obvious that they have to have mitochondria also, right, in order to do aerobic respiration, right? The acetyl-CoA, the citric acid cycle, et cetera. All right, so let's take a look at this image zoomed in on one of these chloroplasts. And even more specifically, we're looking at one of those little stacks of membranes, this little thylakoid membrane stack here, right? This cartoon should kind of ring a bell because it looks a lot like the cartoon of what's happening inside of a mitochondria, right? Remember, in the mitochondria, pyruvate comes in, it's converted to acetyl-CoA. That acetyl-CoA feeds into the citric acid cycle, right? And then you get all these electrons off of the carbon-carbon bonds that came in originally from the glucose that you're oxidizing. And then those electrons, right, what do? They power an electron transport chain that pumps protons across the membrane of the mitochondria so that you build up a strong proton gradient that is then allowed to flow back downhill across the membrane through special ATP synthase complexes like the turnstiles, right, that a big crowd of sports fans push through to get into a stadium, right? They're hooked up to generate lots of molecules of ATP. So we see that here as well inside of this chloroplast. So to keep it simple to follow, let's start from this part here, the production of ATP, right? This part looks exactly the same as what we saw inside mitochondria, right? We're going to have a strong proton gradient that's going to be flowing protons through this membrane, right, from one sort of chamber or compartment of the thylakoid, right, to the other side. And by flowing through there, the turnstile turns and you make 3 ATP molecules per turn, right? That part's exactly the same as what we looked at before. Let's trace this back. Where did this strong proton gradient come from? Well, it came from an electron transport chain or electron transport system, right, that was pumping those protons across the membrane to build up this strong proton gradient, right? And how does that work? It has to do with electrons flowing through these big ETS protein complexes, right? And as these electrons flow through, right, protons get carried with them. I don't know if I did a good job of explaining that last time, because I think a couple students have asked, how does the flow of electrons through ETS protein complexes result in the pumping, quote unquote, pumping of protons across? Some of you have asked, is that like active transport like we learned before? No, it's not quite the same as active transport, right? Remember, active transport is when you have proteins in a membrane that are in charge of moving some kind of solute molecule, right, across that membrane from one side where that solute is at a low concentration to this other side where the solute is already at a very high concentration, right? And in order to do that, this protein, this, like, for example, active transporter has to change its shape in a way, right, to actually push or shove the solute across. We said that requires energy because normally the shape of this membrane, right, would want to stay or the shape of this protein in the membrane would want to stay a certain way because you have so much solute on this side, basically discouraging it from changing the other way, right? So you'd actually have to bind something like ATP and use some of that energy of the ATP to actually force the protein to change its shape against the force of the gradient. And that's how you shove another solute molecule across, right? In electron transport proteins, right, it wouldn't really make sense if you have to use ATP up to do this, to move protons, because the whole point of this is you're trying to make ATP, right? No. So in those special protein complexes, the movement of electrons through there, right, basically carries with them protons. Because remember, a proton is just a proton. It's just like this positively charged molecule. And then you have an electron, right? A proton and an electron would love to come together and be hydrogen, right? So if you have electrons that are being moved through a pathway, then protons will sort of automatically, happily join up with those electrons and be carried along that pathway as well, until you get to a certain point when those electrons maybe move on to the next complex. The proton breaks off and falls off, but it falls off now being on the other side of the membrane. All right. So these electron transport complexes in photosynthesis are very similar in principle to the ones that we looked at in the mitochondria. They're going to move electrons through them, right, to carry protons across to make that proton gradient, right? But a big difference is, where are these electrons coming from? Remember, in the mitochondria, you have all of these electrons that are being fed into the electron transport system from all of these NADH and FADH2 electron carrying baskets, right, that were filled with electrons from things like the citric acid cycle or from previous steps of aerobic respiration, like the conversion of pyruvate to acetyl-CoA, or even back in glycolysis, when you have the production of or the filling of a basket during anaerobic glycolysis, right? So all of those baskets dump electrons into the electron transport chain of the mitochondria, and all of those electrons came from a glucose molecule, right? So here, right, we're not talking about using glucose as the source of electrons, right? We are actually going back, right, to square one, right? Starting from scratch, getting energy from sunlight to be the power that provides electrons to go through the photosynthetic ETS chain. Where do the electrons actually come from? They're not carried by the light. The light is just the energy to move these electrons. These electrons are coming from the breaking of water, right? When an H2O molecule is broken and the two electrons come off, right, those two electrons go into the photosynthetic electron transport chain. And it is the energy from light that's breaking them off of the water. Once these electrons are removed from water, what are you left with? You're left with the O2 from the water, right? And that's where the O2 as a waste product of photosynthesis happens, right? So this O2 is then released by the plant into the atmosphere. These electrons then flow through, right, and set up that proton gradient. So critical to understanding then how this photosynthetic system works is understanding how light is harvested, right, and used to actually motivate electrons to start moving through. Right? So today we're going to focus for the rest of this lecture on what's going on up here. Right? Next lecture, we'll take a look at the second part of photosynthesis, which is the production of glucose down here. All right, so let's take a look at these ETS molecules and what's going on. So these big electron transport complexes, just like in the mitochondria, they move electrons through them with the help of special prosthetic group like molecules, right, that are shaped or designed to do a very good job, right, of transmitting, in this case, energy. So chlorophyll, that's what we're talking about. So this is the shape of chlorophyll over here, right? And here is its structure. So chlorophyll is the two main important types of chlorophyll that are involved in green photosynthetic plants are based around an ion of magnesium. And then around this magnesium, you can see there's this complicated, almost like a net-like structure that's made, right? And there's all kinds of double and single bonds in it. The overall three-dimensional shape looks a lot like a net, right? Something that you might catch bugs with, or, you know, as a kid, if you like going to the tide pools and you're sort of trying to catch the fish in the tide pools, right? And that's exactly what this is. It is a molecular net, but what is it trying to catch? It's trying to catch sunlight, right? So the two main types of green chlorophyll are chlorophyll A and chlorophyll B, right? So this is their structure here. The only difference is this particular R group up here is what's different between A or B. On the left is a graph showing that these chlorophyll molecules have different sensitivities to different wavelengths of light. So remember, light is a series of energy in the form of waves, right? And so the wavelength of light can be measured in nanometers. The smaller the wavelength, right? That means the sort of more narrow the actual waves of these light are, right, would be things like the purples and the blues. Right? So down here, this rainbow shows you the actual wavelength of visible light, right? What we can see with our eyes as being colors. Right? So the tight, small wavelength, waves of light, right, are down in this range. If you get smaller than around, let's say, the 400 nanometer range, our eyes can no longer see that as color, right? So that's no longer violet. That would be ultraviolet, right? Ultraviolet light that's coming as part of sunlight that we don't actually see as color, but boy, we sure know its effects. Right? As the waves get wider or longer, right, you get more into the yellows, then oranges and reds. Okay, so what you see here is you have different types of chlorophyll, like chlorophyll A and chlorophyll B, which are shown by these two green lines here. And this graph is showing you what particular wavelength ranges are these chlorophyll A and B molecules particularly sensitive at being good at taking sunlight and converting that into energy. And what you see is if you just look at where the light green and the dark green lines are, right, because that's the chlorophyll A and B, you'll see that chlorophyll A and B are most sensitive to light in the blue range, okay? And if you follow them along, right, they are also sensitive to light in the oranges and the reds, okay? And that is because the particular structure of this molecule means that it'll work best, right, when agitated by only certain frequencies of waves hitting them. So here's a question. Why is chlorophyll A and B green, right, when we look at it? Because when we look at a green leaf, we see green, right? And that's the chlorophyll A and B. It's because look at its sensitivity over the green range, right? So here's the green color. You can see that chlorophyll A and B do not absorb green wavelengths very well at all. Does not convert green wavelengths into energy. What happens to those green wavelengths? They just bounce right back from the molecule, right? So when sunlight hits a leaf, right, we see it as green because the chlorophyll has absorbed the blues, the oranges, and the reds, but the green wavelengths aren't absorbed and they bounce back to our eyes. So the leaf looks green to us. Okay, because, so in other words, the answer is the green is not absorbed and it bounces back, and that's what we see. Right? So let's think of light. Light as packets of energy. So you might know from physics, it's kind of weird. Light has this weird property of being both waves and particles. Right? So I already said that shorter wavelengths equal higher energy. Right? So you can see here, these waves that are tightly packed together, right? That would be like the violets or even the ultraviolets. Right? That's energy that's high enough to actually damage your cells. Whereas waves that are broader or wider is lower energy. And that's when you get into the reds. And when you get too wide of a wavelength, we no longer see that as color anymore, right? But we feel it, we feel the energy as heat. Right? So that's like infrared. So consider this. It's one thing to think about energy as waves or light energy as waves, but what does it mean to think of it as a packet, right, or particle? That's because these waves travel as sets. So those of you who are surfers, right, or just anybody who likes hanging out at the beach, here in Hawaii, we're very familiar with it, right? Waves come in as sets. Right? So you can think of each particle of light as a set of individual waves moving together as a discrete photon, right? So that's the particle nature of it. But within that photon, right, the wavelength of the light in that packet, right, has to do with its energy. So if you're like 25 yards off from shore and you're just kind of like floating around in the water having a good time, you know what it feels like when a big wave comes. It just kind of lifts you up and puts you down, right, on its way to the beach. And that's because the wavelength is very wide at that point, right? So that energy that you feel is much lower because it's basically spread out. But that same wave, as it gets closer to shore, what happens, right? As the bottom of the ocean gets shallower and shallower, that energy is forced upwards and concentrated, and the wave becomes more compact. So that same wave, if you are now in the crash zone, like just maybe like 3 yards from shore, that energy is much stronger and can topple you over, right? So that has to do with the energy having to do with the wavelength. All right, so we all know that sunlight is comprised of light of all different energies, right? So across the entire spectrum of visible as well as invisible light, all of that is contained in sunlight, right? The white light of the sun. So that's why if you're able to break up those wavelengths and get some to travel in a slightly different direction than others, like through refraction, that's what rainbows are, right? So rainbows are the white light that is now split up because the light waves are bouncing off of things like the water particles or the different surfaces of the water particles, refract the light and change their direction so we can see the colors come out, right? And each one of those represents different energies. So when you look at a chlorophyll molecule, right, here we have the chlorophyll molecule, different photons of light then strike this molecule, right? And the energy will excite the electrons. Right? So if this were low energy, that is not visible light, but just heat, right? You can think about it as if you heat up, for example, a cup of water, right? And all the H2O molecules themselves begin to vibrate, right? And the more they vibrate, the hotter the water is, right? But imagine if that's just heat energy, right? What if you actually get up into the visible range of light? Then that energy is high enough that you're not just moving the entire molecule, you're actually beginning to really mess with the electrons in the bonds in the molecule, right? And that's what happens. So the electrons that are part of this chlorophyll molecule, they themselves absorb the light. And if those electrons then change their energy due to absorbing, this photon of light, what happens? It changes their ability to engage in bonding. So in this chlorophyll molecule, these single and double bonds that are part of the structure here begin flipping around, right? So they start moving around. What was single bond then becomes double bond, then it flips around again. And that represents this molecule of chlorophyll absorbing this wavelength of light. So think about it this way. Right? The structure of this molecule, right, is fixed. And as a result, that's why it's only sensitive to certain wavelengths. It's like if you have a glass of water and you were to put it in front of a really loud loudspeaker, and then you somehow were able to sort of blast out a range of different pitches, starting from super low bass pitches all the way up to treble, right? I think you would know that as you go through the different frequencies of sound, right, you would see that the water at some points would begin to sort of ripple, right? And that's because that happens to be the right frequency range, right, that resonates within this glass, right? And those frequencies would be dependent upon the shape and size of the glass that you have. In other words, the structure of the molecule would define its sensitivity to what ranges, in this case, light, this molecule is sensitive to. And that's why chlorophyll A and chlorophyll B, right, its structure or their structures make them sensitive to absorbing light in the blues and in the reds, the oranges and reds, but not in the greens. Okay. And so you might say, well, that seems kind of silly, because why didn't life evolve to have a particular structure, chlorophyll, that's equally good, right, at absorbing all frequencies? And again, because that's not very likely to have one structure that would be equally sensitive to all frequencies of light, right? So early life had to basically pick and choose early on what molecular structures would be the most useful. So why do you think blues and reds became very sort of the desired sensitivity? Right? I think the answer is actually kind of obvious. It's so obvious that we don't think about it. Right? When white light comes through the atmosphere, right, the atmospheric gases begin to scatter some of the wavelengths, right? So the blues get scattered, right? And that's why there's a lot of blue wavelengths that end up bouncing around all over the place separately. And that's why we see the sky during the daytime as blue, right? The rest of those energies, like the greens and yellows and reds, right, they come down just as white light. But what about in the evening or in the morning? What light is being bounced around the most up there? Right? That's when we see more yellows, oranges, and reds. So therefore, right, at those certain times of the day, it made sense that early life wanted to choose molecules that were particularly sensitive at being able to get as much of the blue light that's bouncing around, right, during the day, and still take advantage of the yellow, oranges, and red lights that were bouncing around in the morning and evening, right? Really neat to think about. Okay, so let's take a close look now at the electron transport chain in this photosynthesis. So if you look here, right, just like when we looked at the ETS chain in mitochondria, the photosynthetic electron transport chain in chloroplasts also consists of these main complexes. We call them photosystems. There's two main photosystems, photosystem 2 and 1. I think, you know, they're actually named kind of backwards. I think it's probably because photosystem 1 was discovered first, right? And then later they discovered, oh, there's another one, we'll call that photosystem 2. And then when they kind of pieced together how they all work together, they realized, oops, right, it's actually photosystem 2 where the light harvesting first takes place, right? So here we go. So photosystem 2 up here is this blue one, right? And then the photosystem 1 would be this green one over here. And then you have this other complex, right, which we call a cytochrome BF complex in the middle. So you can see the lightning bolts show where light can be harvested. Right? So when chlorophyll molecules harvest light, we're saying that the electrons in the chlorophyll begin to get excited and the double bonds begin to flip around in there, right, representing the energy from the sun now being absorbed by this molecule. How does that get transmitted? Right? So it gets transmitted through a process known as resonance transfer. The chlorophyll molecules are arranged as sort of like an array of solar panels. You can think of them that way, right, within one of these photosystem complexes, right? So the proteins within this photosystem complex are holding onto these baseball gloves. And in this case, these baseball gloves are the chlorophyll molecules, right? So when a photon of light hits one of these chlorophyll molecules over here, right, the energy is going to go bouncing around in it in the form of the electrons, double bonds, single bonds flipping around, right? But the electrons themselves don't leave that first chlorophyll and jump to the second. Instead, it's almost like this first chlorophyll molecule crashes into the second. And so the energy is kind of transmitted that way. And that's what gets the second chlorophyll molecule, right? Its electrons start being agitated and flipping around. It's just like, you know, this toy, this sort of like kinetic, I don't know what they call it, Newton's cradle or whatever, where you have the steel balls, right? If you drop the steel ball from the left and it hits over here, I think you all know the steel balls in the chain don't actually change their position, even though energy is being transmitted through them, right? And then it's only the steel ball at the very end that has nobody else to kind of crash into. That one ends up being kicked up. Right? So it's the same thing in this photosystem. So the energy from the sun through a photon excites electrons in this first chlorophyll that got hit, right? And that energy is transmitted through a chain of chlorophyll molecules through resonance transfer until you finally get to a special chlorophyll molecule that's in a particular place. And in that case, its electrons get booted off. And those are now the electrons that move through the electron transport chain. Okay. So here is a more detailed diagram of what's happening. So here we have photosystem 2. Right now it looks like a big purple bell pepper, right? But whatever. Okay. And it has all these chlorophyll molecules in it, chlorophyll A and B. When a photon hits this particular chlorophyll molecule here, the energy is transmitted through resonance transfer until it reaches the core of photosystem 2. And there's a special chlorophyll here called chlorophyll, the P680. Right? That's the chlorophyll molecule that ends up coughing up a pair of electrons. Okay. And those electrons then get carried by a special carrying molecule known as plastoquinone, or QB, right? Which now is reduced temporarily by carrying those electrons. And when it moves out of photosystem 2, right, it has these negatively charged electrons. Oh, protons that happen to be up here floating around in the stroma on this side of the membrane happily bind up to those electrons, right? That's how I said electrons end up just kind of dragging protons along. This QB then becomes QBH2, right? Because it essentially has now, right, 2 protons that got added to the 2 electrons that it's carrying, right? So now that's basically like it saying it has 2 additional hydrogen atoms on it. And that carries it to the cytochrome BF complex, right, where the electrons keep getting transferred through, right? But the protons now fall off, and the protons are now falling off on the other side of the membrane, contributing to the buildup of a proton gradient, right? So if this P680 chlorophyll gave up a couple of its electrons, it's not technically the same molecule anymore, right? Because it's short 2 electrons. So it needs to have those electrons replaced. Where do those electrons come from? Those electrons come from the breaking of water, as I mentioned earlier in the lecture, right? So a molecule of water is broken to replenish the pair of electrons that this P680 molecule of chlorophyll had basically coughed up, right? And so that's why you constantly have water being broken here, releasing O2, right, as a waste product that goes away, and that keeps the steady supply of electrons going, right, through this P680 molecule. So this poor P680, right, it keeps getting another pair of electrons from water, but then it's like it keeps getting this boot kicking it, right, in the form of all this energy coming through, trickling through all these chlorophyll molecules through resonance transfer, right? And because, you know, even though the cartoon shows, oh, this is one photon hitting this one chlorophyll molecule, right? That's oversimplifying it. The reality is, right, you'd have potentially, right, hundreds of thousands of chlorophyll molecules within this photosystem, all being hit with photons of light, right? All sending resonance energy through there. So this poor P680 chlorophyll at the center, it can't get a break, right? It's a steady stream of electrons being kicked off of it, right? And that means a steady stream of electrons have to be put back on it from water, right? Okay. And so the result of this is the buildup of that proton gradient that is then going to flow back through the membrane through special ATP synthase complexes to then make ATP. Right? Okay. All right. So this last slide for today is just different ways of kind of visualizing this, you know, because the big purple bell pepper in the previous slide, you know, that only kind of gets you so far in terms of appreciating, right, what's actually happening inside of these molecules. So here are other representations of what these protein complexes look like, right? So for example, I think the most useful one here is probably in the middle. Okay? So this photosystem is made up of all these different protein subunits, right? And these protein subunits are hanging on to, right? They're all wearing baseball gloves, right? These baseball gloves are the chlorophyll molecules acting as prosthetic groups to facilitate transfer of energy. And so here, this lightning bolt says that this particular photon hits one molecule of chlorophyll in this subunit over here, and that sends, right, this energy rippling through all of the single and double bonds of the chlorophyll molecules through resonance transfer, right? So this is the steel ball came and hit this chain of other steel balls, right, in your little desktop, toy or whatever, right? And they're not giving up electrons to each other. They're just transmitting the energy through that chain until you finally get to the core of the photosystem, where you would have that special P680 chlorophyll that actually ends up kicking out its two of its electrons, right? And then on the right here, right, this is a simplified version to remind you that as that P680 gives up a pair of electrons, it has to have that pair of electrons replaced. And those electrons come from the breaking of water, right? So this continuous stream of electrons essentially coming from water going through the P680 molecule, right, and entering the electron transport chain. And what keeps this flowing, right, is the constant stream of energy from the sun, constantly barraging all of these chlorophyll molecules, right, to send this resonant energy down to the core to keep booting off the electrons. Okay. All right. So, I know that sounds kind of like really crazy, right? But at the same time, hopefully you find it really neat too, right? So photosynthesis doesn't have to be this like, oh, this is totally irrelevant, right? It actually ties in really well with a lot of the concepts that we've been studying so far, right? And as life science students, right, it's all about trying to understand and appreciate, right, how the biology of something like plants and photosynthetic organisms, right, is essential to our biology, right? Because ultimately, this plant that is doing this, right, its ATPs are going to be made into the sugars that we depend upon for our survival. Right? Okay. All right. So next time we'll take a look at the second part of photosynthesis, which is how the plant takes some of these ATPs that it's going to make and turns it into sugars. Right? All right. Let's go to the chat if there's any questions here. So yes, so a question is, since plants also have mitochondria, right? So I never want you guys to forget that. Right? Eukaryotic plant cells have mitochondria in addition to having the chloroplasts, because if they're going to make sugars, right, as a way of storing the energy of some of the ATPs that it's making, they have to have a way of oxidizing those sugars to get the ATPs back out, right? And that process is exactly the same as what our cells do, right? So yes, in the same way, the plants would also release carbon dioxide as a waste product. So that's a very good question, because we normally, right, when you talk about photosynthesis, when you learned about it in high school or even general bio, it's oversimplified, right? Plants take in carbon dioxide and give off oxygen, right? That's what we learn, right? And then we breathe in the oxygen and we give out carbon dioxide. And that's why, it's a wonderful cycle, right? But that really oversimplifies things, because at the same time, if plants are going to be using the glucose that they make to get the ATP back out, right, they're going to be doing so through the conversion of glucose to pyruvate through glycolysis, and then acetyl-CoA, the citric acid cycle, and then using the electron transport system in their mitochondria, right, to make the ATP, get the ATP back out of the sugar. And as a result, yes, absolutely, plants also release carbon dioxide, right? But one way of thinking about it is, during the day, if there's plenty of ATP being made by the plant due to plenty of sunlight, right, its production of sugar, right, even though it's making the sugar, right, it may not necessarily be needing to actually use that sugar as much, right? But at nighttime, when there's no sun to make ATP directly, and the plant has to rely upon those sugars that it made during the day, to now break those down through oxidative respiration or aerobic respiration, that's when you would have an increase in carbon dioxide released from plants. Okay? So we'll talk about that next time when we sort of complete this picture by adding on the glucose synthesis pathway after this. Okay, all right, so no more questions. All right, okay, so have a good rest of your Wednesday, and we will talk more about this on Friday. Okay, all right. Bye.